Hello everyone. So today we from group 2 are going to present to you our drama video titled Watson, Sherlock and the Stupid Detective. This is going to be a hilarious drama, so I hope you'll enjoy watching it. And in this drama, we would be illustrating to you what is microbial forensics and how can it be used in real life cases. As for the cast, we have Ku, who is going to be our dumb detective. Adriana will be playing the role of Watson. Shania will be our Sherlock. Chua will be playing the role of Suspect 1. Haida will be Suspect 2 along with the victim. And myself, Pritisha, I will be your Dr. Pritisha, or known as the lab person. So once again, I hope you'll enjoy the drama and yeah, let's go.
No.
Another day with another crime scene. It's clean, no footprint, and no fingerprint at all. This guy must have been wearing gloves and no CCTV. For real, what a perfect crime scene.
Sherlock, police got the suspects. There are two of them.
Alright, let's go see them.
So, two of you are suspects of this murder case. Let's start with your alibi. Chua, where and what were you doing during last night?
Sir, I seriously don't know why I'm one of the suspects. I'm innocent.
Shut it. That is your concern. Give me your alibi.
I was at home around last night. Since today is a weekend, I was just Netflix and chilling.
And you want to prove that?
I was home alone.
And you, hey dear, what were you doing?
I was at home also. Just doing a regular routine of yoga before bed and I was alone too.
So, both of you were at home last night and never left your house. No witnesses?
Yes.
This is headache.
Hello, Detective No Brain. I heard you got the suspects.
Sherlock, language. Good morning, sir.
It's you again? Shush! There are enough problems kicking my ass already. And good morning to you too, Watson.
Why? Wait, let me guess. Is Detective too dumb to find any evidence in the crime? Or is Detective too naive to believe whatever the suspects are saying? Ha ha ha.
You, if you are here to disturb me again, get the hell...
No, Detective. I am here to give help to desperate and dumb detective. I don't have to tell you that desperate detective is you.
Ignore him, detective. It's Sherlock.
Yes, the mightiest genius private detective Sherlock.
Fine. As you said, there are no evidences. No footprints, fingerprints, DNA, and both of suspects say they were home only and never left.
Never left their house, huh? Watson, can you do me a favor?
Yeah, sure.
Go get me their shoes.
Shoes? Wait. For what?
Shh, detective. Just let me do my thing. Alright?
Here you go.
Yes. Wait. Sherlock, can you tell me what's going on?
Later.
Dr. Pritisha, you've got some work to do. It is very urgent.
What the? Just do it. It is for murder.
Alright, give it to me. Interesting. This mud from lakeside has protobacteria and some algae blossoms in it. Here you go.
Fascinating.
And this is more interesting. Some protobacteria and algae from the shoes.
so she must have visited the crime scene.
No, I'm innocent.
Hello everyone, we are Group 2 and the topic we are going to talk about is microbial forensics. Before we start, let me introduce you to our group members, Adriana, Shania, Pritisha, Chua and Co.
This video we would cover about what is microbial forensics, the importance of microbial forensics, the application of microbial forensics, when was microbial forensics discovered, the important cases of microbial forensics, the impacts of microbial forensic advancement, the recent advances in microbial forensics, the threats, the limitations, and the conclusion. First of all, we are going to discuss what is microbial forensic. It has been described as a scientific discipline dedicated to analyzing evidence from a bioterrorism act, biocrime, or inadvertent microorganism or toxin released for attribution purpose. If you explain it in a much simpler manner, it means the investigation of the use of a bioweapon, accidental release of a dangerous microorganism, or the natural development of harmful microorganisms. It also includes studying a person's specific microbiome to identify the person, time of death, and the location of the crime of an individual based on the development of the microbiome during decomposition. There are few importance of microbial forensics. First is to detect and prevent bioterrorism acts. Second is to assist our understanding in what may have occurred during a biothreat event. Third is to support suspected or known bioterrorism investigations by providing investigate leads and supporting prosecutions or exonerations with scientific evidence. Fourth, it seeks to produce reliable conclusions to protect the public health in the case where a harmful microorganism is released. Lastly, it also helps in agriculture and medicine since agriculture goods are susceptible for microbial intrusions, so therefore microbial forensics could be used to detect it.
Application of microbial forensics. In a crime scene, there are plenty of microorganisms laying around in the surrounding area and objects, and in and on the body of the victim. These microorganisms can be collected and used as evidence. The evidence collected is examined using processing tools, redefined tools, and computational tools that are being developed alongside new approaches in bioinformatics. 6 most comprehensively researched themes under application of microbial forensics are 1. Geolocation, and an example is substrate analysis. 2. Personal identification. 3. Biological sex determination. 4. Trace evidence. 5. Manner and cause of death. And 6. Other applications and examples could be localization through animal microbiomes. When was microbial forensics discovered? Microbial forensics only emerged in the early 2000s, and this was due to the rise of bioterrorism. The FBI of the United States Hazardous Material Response Unit established this field of study called microbial forensics in the 1990s.
Important cases. One of the important cases is the anthrax letter attack. It is caused by the Bacillus anthracis, which is a gram-positive, root-shaped bacteria. 5 were killed and 17 sickened. It is transmitted by powdered anthrax spores that were put in letters. Impacts of microbial forensics advancement. First, for forensic identification or human identity testing. Second, to uncover the identity of biological threat. And third, for study of infectious disease.
Recent advance in microbial forensics. Applications of microbiome in forensics. Data generation, individual identification, geolocation inference, PMI estimation, metagenomic analysis. 1. Data generation. Two main methods for characterizing the microbiome. Amplicon sequencing, most popular NGS technology for forensic microbiome analysis. Target marker genes, regions of interest. 16S rRNA gene used for profiling prokaryotic microorganisms and internal transcript spacers. 18S rRNA gene used for profiling eukaryotic microorganisms, characterized by high efficiency, low cost through application of 18S rRNA gene and ITS gene, can be applied to low biomass specimens without affecting host DNA. For an example, Illumina MASEC, most widely used platform for microbiome analysis. Shotgun metagenomics, datasets used to identify SNP markers, provides functional gene information and strain level resolution rather than just taxonomic composition, as determined by amplicon sequencing. Disadvantages: method more expensive, performs poorly on specimens with low biomass or heavily contaminated by host genomes, therefore limited in application to forensics.
These are the threats of microbial forensics. Firstly is the lack of standard procedures for the collection and analysis of samples. This is due to how microbial forensics had only emerged in the 2000s, making it relatively new compared to other forensic fields. Next is the accessibility and cultivability of the microbes that can be easily assessed and cultured by anyone. Thirdly is the capability of large-scale production of the microbes, which will be a biohazard, leaning towards a possible bioterrorism. The ability to retain potency during transport and storage is also a threat towards microbial forensics, as well as the ability for dissemination. Moving on is the incubation period, which is the time from when the infection occurred to the onset of signs and symptoms or the first positive test. The most serious threats, however, are the lethality of the microbes and the pathogenicity with the toxicity and, most importantly, the transmissibility.
Limitations in microbial forensics. Microbial forensics is still in its early stage, and there is so much more to be discovered. Even though microbial forensics involve techniques and methodologies from basic laboratory science, the issue questioned have particular aspect outcomes. Due to such high expectations, there is more to be developed in the aspects of microbial forensics, but regardless, the future of microbial forensics looks quite promising. Conclusion: in general, microbial forensics is associated with microorganisms being detected in a scene of crime or in an act of bioterrorism. Slowly but steadily, the field of microbial forensics keeps developing. Microbial forensics has proven to be quite helpful and will continue to be helpful with the ongoing development of technology.
Oh, wait. Hold on. Before leaving, we would like you to try our recap questions. First, give two importance of microbial forensic. Second, describe what are the limitations of conducting microbial forensic. Third, is microbial forensic efficient? Justify why. Fourth, discuss the advantages and disadvantages of microbial forensics. Fifth, briefly describe a scenario where microbial forensic is applicable.
Thank you so much for watching. Do give us like and subscribe. Do share to your family, friends, colleagues, or the people around you to let them know the importance of microbial forensics for a crime scene.